Good morning. Welcome to Currents Chem World. Let us see today chapter-wise chemistry quiz in biomolecules. These are the important questions asked in various competitive exams. Let us see the first question. Raffinose on hydrolysis gives. Raffinose on hydrolysis gives. It is a trisaccharide. On hydrolysis, it gives glucose, fructose, and galactose. The second question is the pH value of the solution in which a polar amino acid does not migrate under the influence of electric field is. Called isoelectric point or isoelectric pH, not isoelectronic. It is isoelectric point or isoelectric pH. At this pH, the amino acid molecules are neutral. At this pH, the amino acid molecules are neutral. Therefore, they do not migrate either to the positive electrode or to the negative electrode. It has the least solubility at the pH. So, at this pH, they have less solubility that can be easily separated, and also for each and every amino acid they will have characteristic isoelectric point. They will have different pH, isoelectric pH values for different amino acids, different isoelectric pH. The protein which controls the amount of sugar in the blood is insulin. Insulin is the protein or hormone which controls the amount of sugar in the blood. Insulin is a protein hormone. The fourth question is riboflavin is vitamin dash, B12 or vitamin B6, B2, etc. It is riboflavin is vitamin B2. Its deficiency causes skin disease. Riboflavin is vitamin B2. Please remember this. Let us see the fifth question. The change in amino acid sequence leads to process called mutation. Mutation is a process in which there is a change in amino acid sequence. Okay, the amino acid sequence gives primary structure. The sequence of amino acids in protein gives primary structure. Any change in amino acid sequence will lead to mutation. For example, sickle cell anemia is caused due to this mutation process. Sickle cell anemia, okay, hemoglobin, that is sickle cell disease due to valine 2 glutamic acid, valine 2 glutamic acid in hemoglobin in red blood cells. Due to this, it cannot transport oxygen. The hemoglobin will be sticky and causes sickle cell anemia. This is basically due to mutation. Mutation is the change in amino acid sequence. Let us see the next question. Glycosidic linkage is an dash linkage. Peptide linkage or polyester linkage. Glycosidic linkage is an ether linkage. Glycosidic linkage is an ether linkage. Let us see the sixth question. Glucose fails to react with glucose has aldehyde functional group, so it will react with the phenylhydrazine. Glucose fails to react with the sodium bisulphite. In general, aldehydes react with the sodium bisulphite, but glucose will not react with the sodium bisulphite. Progesterone hormone is called pregnancy hormone. Eighth question. ATP stands for adenosine triphosphate. It is also called energy currency. ATP is called energy currency. Energy is produced in our body in the form of ATP. Ninth question. RNA consists of pentose sugar. RNA consists of pentose sugar. 5-member ring, ribose pentose sugar. 10th question. DNA consists of pentose. DNA consists of pentose. Deoxyribose. In RNA it is ribose. In DNA, deoxyribose. 11th question. Vitamin dash is synthesized in skin cells in the presence of sunlight. What is the vitamin? It is also called sunshine vitamin. Vitamin is synthesized in skin cells in the presence of light. Vitamin D is called sunshine vitamin. It is also called calciferol or ergocalciferol. 12th question. Vitamin dash is essential for blood clotting. If this vitamin is deficient, blood won't clot. Blood loss will lead to death. What is the vitamin? Is essential for blood clotting, blood coagulation. Vitamin K. It is vitamin K. It is also called anti-hemorrhagic vitamin. Its chemical name is phyloquinone. 14th question. Dash structure gives three-dimensional arrangement of proteins. Dash structure gives 3D arrangement of proteins. Tertiary structure. Tertiary structure gives three-dimensional arrangement of proteins. Dash structure explains how the subunits are spatially arranged. Dash structure explains how the subunits are spatially arranged. It is quaternary structure. For example, hemoglobin has 4 subunits. There are 4 individual subunits. Okay, how they are arranged. It is explained by quaternary structure. Each subunit 3-dimensional arrangement is explained by tertiary structure. Alpha helix and beta sheet structure corresponds to dash. It corresponds to secondary structure. Secondary structure explains the alpha helix beta sheet structure. It also due to conformation and also gives backbone chain. The conformation is also due to secondary structure. 17th question. Human body can synthesize vitamin A from dash. Human body can synthesize vitamin A from dash. Carotene. Carotene is present in carrot. It is converted into retinol. It is provitamin. It is called provitamin. Carotene in carrot is converted into retinol in our body. Its deficiency disease is night blindness. You won't see in the night. Let us see the next question, important question. Zwitter or dipolar ion is exhibited by amino acids. Amino acids in a same molecule have both positive ion and negative ion. Amino acids in the same molecule have, because they have two functional groups, amino acids. 19th question. Optical inactive amino acid is. Which amino acid is optically inactive? It is glycine, which is optically inactive because there is no asymmetric carbon atom in glycine. 20th question. A dipeptide called dash being 100 times sweeter than sucrose. It is called artificial sweetener. Aspartame. Aspartame, it is a dipeptide made of aspartate and phenylalanine. It is 100 times more sweeter than sucrose. So it is used as artificial sweetener. 21. Dash amino acids cannot be synthesized by our human body. Give some examples. Essential amino acids. Those amino acids cannot be synthesized by our human body are called essential amino acids. Non-essential amino acids can be synthesized by our body. So essential amino acids cannot be synthesized in our body, such as tryptophan, phenylalanine, histidine, arginine, methionine, etc. Let us see the next question. It is very, very important. It is asked in all the competitive examinations from NEET to GATE CSIR. The question is, how many hydrogen bonds are present between guanine and cytosine and adenine and thymine in DNA? Guanine, cytosine, adenine, thymine or nucleic acid bases in DNA. As you see, adenine pairs with the thymine. Adenine is complementary to thymine, so it forms hydrogen. Adenine forms two hydrogen bonds. Adenine forms two hydrogen bonds with the thymine, whereas guanine is complementary to cytosine in its structure. So guanine binds with the cytosine to form three hydrogen bonds as shown here. GC pair with the three hydrogen bonds, AT pair with the two hydrogen bonds. GC pair is more stable because of three hydrogen bonds. It is very difficult to break. it can be easily broken. Let us see another important question asked in NEET 2 GATE CSIR. The complementary strand for the following single strand of DNA is. What is the complementary strand for the following strand? Single strand. Okay, what is the, it is DNA is a double helical structure. It is one of the strand. From this strand we can predict the complementary strand by using this adenine pairs with only with the thymine. Okay, now this is the answer. Let us see how it is obtained. So this is the strand. Okay, we need to build complementary strand. So first let us see, adenine is complementary to thymine. So this is present in the complementary strand. Next, thymine is complementary to adenine. So the next one, guanine is complementary to cytosine. Therefore, next, cytosine is complementary to guanine. Next, thymine is complementary to adenine. So 5 prime to 3 prime means 3 prime to 5 prime. So this is the single strand, this is the complementary strand we have obtained from this. Thank you for watching. Please subscribe Current Scramble and watch for more videos like this. Thank you.